Survey says that really interesting breakdown. 50% want the exam on Friday, February the 21st, and then the other two are pretty close at 22 and 27%. So Friday the 21st, which is good because I'm not going to be, I don't think I'll be here that day, so that'll be a good day for you to have the exam. And I'll send that out later. Okay, let's go and look at the quiz.
Oh, when you were saying about the graphs, you said them as active. Will it mess up if we leave the test?
You'll have it in your hand.
Oh, okay.
I mean, you'll print it out.
Oh, okay.
Okay, if I do that, yeah. So, yeah, that's a question. I just screwed up. I did not have my pop-up blocker off, and I missed a screen. And there's something called a demo student, so I can go to the student view, but it only lets me view it if it's active. So I have to build a test, then go view it, and then come back and add the time. You know, and that was just my fault. Most of you did take it though, and you're getting the right feedback, and so. Can you guys see that? We are recording, right? Okay. This is only composed of glucose. Which one of those? Cellulose. Survey says cellulose. This is the kind of cell wall that is found around all plant cells. The primary cell. The primary cell wall, let me tell you, it's outside of all the other cell walls. Okay? So you've got the primary cell wall, and then the protoplast. And then if secondary cell walls are going to be made, then the Golgi, that entire intermembrane system is going to start pumping stuff out to the cell membrane and dumping it outside as the cell thickens from the primary cell wall in. Okay? This is the region of the plant where division occurs. Meristem. This tissue has secondary cell walls and conducts water in the plant. Xylem. This is a question that we formulate based on our observations. This one. I'm sorry?
How is that a question that you formulate? I mean,
This is a question, the question that we formulate is the hypothesis.
that's what it is. I put that in
An experimental design.
Okay, well then I made a mistake. Like I said, when I do these, I'm going to make mistakes. So, and then if I do, I can actually go in there and change the individual questions. Yes, ma'am.
Question number 3 is, what is the only elongation operative isn't that where the root lies?
No, the only place there's division is either in the root or shoot meristem. And then both in the root and shoot. And then in the root, you even see a much more pronounced zone of elongation. The root, you don't see much of the little red, real thick red stuff where the divisions occur. There could be some divisions in the zone of elongation, but almost 99.9% are going to be in one of the meristems. And that one said experimental design. Okay. Also, I'm not going to reduce your grade when I make a mistake. So I'm not going to go back and regrade and change. So if you know what I'm talking about. The variable that we control, the independent, it means independent. Lipids have this characteristic. They are hydrophobic. Not lipids.
But the membrane is made of the lipid bilayer.
Right, which is a phospholipid, where one end, the phosphate group, The phospho end is basically water soluble, but the lipids themselves are the part that are hydrophobic. That would be what you'd probably call a trick question. I didn't mean it to be. Where the phosphates are the hydrophilic portion. Cell wall that gives rise to thickening, of course, is the secondary cell wall. You would find lignin in where? The secondary cell wall. Now, in a question like that, where you said, well, that's something you could argue about, where you said that you would put both. That's a question you definitely could argue with me about. Okay, and I'm just looking right here. Somebody would come up and say this portion of the membrane would contain phosphate. You could probably come and argue and show me an example of a protein that's been phosphorylated. But I would not. So when I look at the results of these, there'll be. We get a tremendous amount of information from an exam. And one of the most useful, now I hope we do online, one of the most useful pieces of information we get back is who missed what. So if I look through and 80% of the people missed this question, then there's a good chance something was wrong with the question. If 80 or 90% of the people got the question right, then that's probably the question. I didn't put the wrong answer down. Then it also looks at the scores versus what you got right and wrong. And the logic being the higher score you make, then your choices, and this is sort of hard to explain, then your choices are going to be more right. So we get a signal on a question where random people got it right. So in other words, if you make a 99 or if you make a 50, the probability of getting that question right is the same, which means that's not a good question. Does that make sense to you? So it's a discrimination factor. Now, most discrimination factors are like high because in most multiple choice questions, the majority of people will get them right. And so we can go back and look, and I can look at that question. And so in other words, the correct answer was correct, but there was no reason why you got that right answer. It was random. And then I can look at that question and say, no, that's not. I can see why people would put this other answer down. Does that make sense to you? So it's the higher. So it looks at your score and what you got right and wrong. And then it does a calculation based on your score versus the average score versus right and wrong. And it comes up with a measure that tells us that's the right question or not. So if it's got a really high discrimination factor, I'm not supposed to use a question again. Well, I always use them again. So I hope we get that information. I'll know after that weekend. Okay? Any other questions before we start up? Okay, let's see, we're still recording. I can close that, minimize that.